All right, to start things off, I'm gonna explain what this class is, and well, people from the previous year, and maybe it is the common requirement for everyone, but people from the previous year, they asked for a more clear system for getting a grade for this class. So it's gonna be an exam, and so to get the maximum grade, which are presented on the screen, so you need to fulfill several requirements, and these requirements are attending and participating in 2 seminars during the semester, well, completing 2 assignments, writing a test, and the test is going to be, yeah, I'm going to tell about it later. And if you fail to achieve some of these points, you can still rectify, so you can still get a better grade by attending an exam and answering some questions. So an exam is, well, it's an oral exam, and you receive a list of 2 questions. So you need to answer both of them. If you, well, if you didn't do a lot of during the semester, maybe missed 2 assignments or missed tests, so you probably need to answer everything to get a good grade. But usually and ideally, I expect that you do not need to answer anything on the exam itself. So first off, about the seminars, and the seminars are going to be very similar to what you experienced in the very first semester of your education in this master's program. So while it is called Biotech Seminars, it's going to be something like a journal club, and the most prominent difference here would be that I will try to get some papers that allow you to reproduce results in them. So I would expect that that you not only do the presentation on them, but you also try and, well, pull from some open repositories the results of these papers, try to use the data that authors made available in some public repositories, and you try to replicate the results. So in different cases, it may be slightly different because, but I'll try to make it achievable. So maybe you will fail to do so, and in this case, the difference from the journal club will be very, very small, but hopefully you will be able to do something new and I don't know, learn to use some new bioinformatics tools in the process. So the second point and the second requirement for the successful completion of the course is completing the assignments, and one of them is, well, I already know what it will be. So the first assignment will be to take any futuristic biotechnology from fiction and to make a presentation on how would you make it happen. So I would expect something actually achievable. So I have a list of things that people used in the previous year. So I have some ideas that you may want to replicate and you may want to explain in your presentation. Question in the chat, space minutes. Yeah, sure. And people didn't do this in the last year, so you have your chance. So even if you replicate something from the last year, it doesn't matter. So I will just send you the description of this assignment at some point later. And there's going to be a second assignment. I'm not secret here means that I'm not sure what it's going to be yet. So I have 2 ideas. One of them is reliant on the hardware. And if it arrives in timely manner, we'll be able to do this. If not, we'll be doing something like a competitive game between 2 teams of you. So either way, it is going to be something. And I'm just not sure yet what it's going to be. And the last part is that we're going to be having a small test approximately in the middle of the semester. And it's going to be something of, well, to test your knowledge on the first part of the course. Probably put it on Stepic as an exam with a limited time. And I'll try to make sure that it's Google proof, so you are free to use whatever you want during this test. So in a sense, it's a competition between you and me, because I would like to learn how to create Google proof questions, and well, you probably want to complete the course successfully. So here's the course structure, and here's how I imagine the schedule for our course. And as you may see here, today is the only day when I'm going to be reading 2 lectures, and from the next class, we're going to be having 1 lecture and 1 seminar on different topics. So here you can see the topics for the seminar, for the lectures. Maybe I will change some of this later, but I'm pretty sure that the picture is going to stay that way, the overall picture. And of course, I'm going to be sending you a syllabus in the Google folder, so you'll see what topics we are going to be covering during the semester. Oh yeah, and the next week we are not going to be having any class because I have to go to another city for a business trip. So you will have 2 weeks for preparation for the first seminar, and I'm going to be sending you papers for this shortly. So yeah, and the last part of the class, of course, and it is not mandatory as the previous ones, because, well, I'm going to be recording lectures, and I hope that we are going to upload all of them on YouTube somewhere, I don't know. So maybe it's not going to be publicly available, but we're going to put some links for this so that you will be able to listen to them even if you are not present during the actual class. So of course, you will not be able to interact with your classmates, but, well, I guess it's whatever for lectures. And so lectures are mostly based on the same plan, is that I will talk a bit about the biological part of some biotechnology topic, and I will provide some application examples, maybe from the recent news, maybe something from more distant past, and also I would like to provide some possible related bioinformatics applications. So we are studying the master's in bioinformatics, so I try to make it relevant here. So it's not just a biological course, so I try to at least provide some ideas on how you can apply your knowledge in bioinformatics, how you can apply your bioinformatics knowledge here, the biotechnology. And yeah, so that ends the introduction part. So if you have any questions, feel free to ask them. So if not, we are just gonna continue to the next part, which is the first lecture of this course. And the first lecture is gonna be covering the basic genetic engineering. And here you can see the approximate plan of this lecture. So you may notice that it is a fairly basic stuff covered here. So I expect that you may phase out during it if you see that the stuff I'm talking about is very familiar to you. It's not a problem, I guess, because during the online class it's just a natural thing to maybe focus on the things that you are actually interested in. So yeah, here's the plan on the slide, and let's talk about the basic genetic engineering. And for this lecture and for basic genetic engineering, I like to think about this text metaphor for DNA that is presented on the screen. And you can see that, well, you probably know that DNA is just a huge amount of text that encodes the genetic information that is needed for our functioning. And so all the basic information transfer processes in the cell, the molecular biology dogma, in the central dogma, they can be replicated within this metaphor. So if you're talking about the transcription, so here it is, it's the way how you transfer the text from one modality to another. In case of a human speech, it is going to be from the written form to the audible form. Now, in case of translation, you can just upload it to the Google Translate and see that well, some of these words, they can be translated into another language with a different alphabet possibly, and well, it produces a slightly different result. So if I hear a phrase in the unfamiliar language, it's probably not gonna produce any effect on me. And if I hear a phrase in my mother tongue, it's probably, well, it's probably gonna be understood by me. So here it is, and using this metaphor, we can actually widen it to some more higher level processes. So we also have a syntax elements such as promoters, enhancers, insulators that make sure that our text is, well, somewhat structured. And you also have the meaning part. So it's not just the, well, nonsense that is written in the DNA text. It's actually some functions are encoded, and there is some obvious connections in the parts of this text. So which are presented in the real life by the gene regulation networks. So if I use this metaphor for DNA, the genetic engineering, in a sense, is a usage of a lost technology that was provided by some ancient precursors, some ancient civilization that, well, they, in works of fiction, they may have vanished, and they lost some things that can only be used in a very primitive way. So we cannot exactly replicate it, but we can use parts of it to achieve some results that are maybe beneficial to our ends. So in case of genetic engineering, it means that we have a lot of, well, parts that can be jumbled together, and that we can achieve, well, some results from this. So we can take a gene that encodes for some interesting protein, you can put it in bacteria, and we can hope that this magic word that our precursors left for us, it will produce the expected result. So in our case, of course, this precursor civilization is, well, the biological life itself on Earth, and we just use the strange technology that is, well, ourselves, basically a building blocks of the organic life on Earth. And so the basic genetic engineering here is just a very primitive way to use these parts that have a meaning, and they can produce some functions. And the way it goes is that the main idea here is that you have some kind of a DNA part that encodes an interesting thing, and you have a vehicle that can accommodate this interesting part of the DNA, target DNA, and this vehicle can be used to reproduce and multiply this target part of the DNA. So of course we cannot actually achieve it outside of the living cell. So we can achieve it, but it is, well, in the most basic case, when genetic engineering was invented, it was only achievable within living cells. Now we can do this in vitro, but it is still cheaper to use this, to just hijack the replication systems within the living cells and use it to our ends to produce some proteins or maybe some other stuff. So when talking about genetic engineering, we'll start a little bit about the visualization of processes, because while we can, well, do all of this blind, but you would want to have some idea on what we are working on. So, and in case of the, well, working with DNA solutions, it may be a problem, because all the DNA solutions, they just look, well, very similar, because it's a transparent liquid basically. So you would expect that well, we would want something to visualize the process that we're working on. And the most basic way to do this is to use one of the natural properties of DNA, that is that it's a negatively charged molecule, and we can use the electrical field to separate molecules of different sizes. And you can do this because well, DNA molecules, depending on their size, they have different mobility in some circumstances. In case you put the DNA molecules in gels of different nature, they may have different mobility because, well, DNA molecules, well, for instance, the long DNA molecules will have a hard time passing through all of this mesh, and the short ones will have no such problem. So, and well, as I mentioned, the DNA solution itself is a transparent solution, so we would want something to highlight the presence of DNA, and in this case, the special compounds are used that in this case are able to multiply the fluorescence of DNA when they intercalate between the two strands of the DNA. Here's one of these compounds is provided on the slide. So in this case, we are able to see that. So if you put it in the gel and if you add the pigment, so you'll see that short fragments of DNA will be farther away than the long ones, so you'll be able to distinguish them when you are maybe working with a target DNA that is shorter than the vector DNA. So there are some modifications to this because the electrophoresis itself, the simplest form of it does not allow you to use to separate very large molecules of DNA, and in this case, you would want to use some special tricks to lure DNA inside the mesh of the gel, and so that you can separate some very huge DNA molecules. So one of such tricks is to use the pulse field gel electrophoresis to rotate the direction of the electric field so that DNA will eventually get into the gel, and it allows you to separate the bigger DNA molecules. And another way to visualize the DNA fragment, and it is more related to the DNA metaphor for DNA, is the DNA hybridization. And the idea here is that DNA can be denatured at higher temperatures. And well, so it means that from its double-strand form, it can be coerced into forming two strands of the two single strands. And in this case, you can exploit the fact that DNA strands, they interact with each other by the principle of complementarity, and you can basically use the generic control F function to find some fragment that is similar to a query. And it is actually slightly better than control F because in case of the DNA hybridization, you're not required to provide the query that is that has one-to-one correspondence with the search database. So you can use a query that is somewhat erroneous. So you can use something like a regular expression to find your DNA fragments. And in case your query is somehow highlighted by maybe fluorescent compound or radioactivity, you can find where your query can anneal to the database of DNA molecules. So so of course it is used as a tool to find some subsequences within the huge mixes of DNA molecules. So you can use it as this in a way that is combined with the DNA electrophoresis. So in this case, you just first of all you separate the DNA molecules in the gel, and then you use the query to find where exactly your interesting DNA molecule resides. And as I mentioned, you would want something to detect your DNA probe after all. So you would want to add some kind of a tracer to this small DNA molecule so that you would be able to say where exactly it is. And yeah, it may be a fluorescent molecule so that you can later just use the light to excite it and to get the light signal. You can use it. You can use some kind of antigen to later visualize it with antibodies, maybe possibly also conjugate it with the fluorescent dyes, and you can also use the radioactive compounds so that you can use autoradiography for the detection. Um, yeah. So the next part is talking about how do we actually get the parts of DNA that are interesting. So we know that the DNA in the cell, it is a collection of huge DNA molecules. So you're probably not interested in most of it at any given time. So maybe you're looking for a gene that encodes some kind of a hormone or maybe an enzyme. So in this case, you would want some tools to actually get this part of the DNA. So, and of course, in order to get a workable amount of this DNA, you would want to also hijack the technologies that was already present in the cell. And in this case, you may use the DNA replication. But the problem is that in the cell, it requires a lot of actors in order to replicate the DNA. Good news for us is that DNA amplification in vitro does not require all of this stuff. So you only need polymerase, building blocks, and energy. And good thing that they are the one and same. You need primers, and you need a single-strand DNA to serve as the template for the amplification process. Also a good thing for us is that while DNA polymerases are very huge protein molecules, and they may be not exactly stable in vitro solution, and well, the most important activities of DNA polymerases can be decoupled from some less important parts of it. And in this case, you can see on the slide the construction of of DNA polymerase 1 from Escherichia coli, and you can see that the polymerase activity does not require one of the subunits from this enzyme. So you can just get this part, and you're just good to go to use it to amplificate some DNA. So yeah, of course, polymerase requires primers because DNA polymerase is not able to synthesize DNA from just one single strand. So in our case, the good thing is that we do not really need an enzyme that synthesizes primers. You can just provide some synthetic oligonucleotides. So yeah, that concludes the DNA synthesis in vitro, and by combining all of this, by fulfilling all of these requirements and combining all of this stuff, you can perform the best function in the text editors to use the Ctrl+C and Ctrl+V to just pick a part from the text and to multiply it any amount of times so that you get a workable amount of DNA for your genetic engineering experiments. So you're probably familiar with polymerase chain reaction, and the killer idea here, the best feature is that people somehow decided to use 2 primers instead of 1. So DNA synthesis was slightly earlier than PCR, and the people only used 1 primer, and it does not lead to the chain reaction, because if you use 2 primers that are facing each other, you'll get a lot of product in the end that is located between these primers. And yeah, so here you can see that the amount of product grows exponentially. So after 30 cycles, you will get a whole lot of this DNA. So yeah, let's talk about the specific parts of this polymerase chain reaction. So first of all, the primers are nucleotides that are specific to some region within target DNA. So if you want to amplify some coding sequence, you want your primers to start with the coding sequence, and the opposite primer is going to be where this coding sequence ends. And they are characterized by several parameters like temperature of melting. It's a temperature when the 50% of this oligonucleotide is not annealed to the DNA template. They are also characterized by length, the GC content, and the secondary structures. And also some additional sequences can be added to the 5' end so that you can use it to add some new sequences to your target DNA. The next essential part for the polymerase chain reaction is the polymerases, and for PCR, it is important that you use the thermostable polymerases because one of the steps in the PCR is the annealing, or not the annealing, is the melting of the DNA, and this melting requires high temperatures for long DNA chains. And in this case, while you heat up your solution to this temperature, normal polymerase from an organism from temperate environment will probably not survive this. And good thing that we have some organisms that actually live in this very non-hospitable environments with high temperature, and their polymerases are able to operate in this very strange conditions. So in this case, you can exploit this polymerases from thermostable organisms for polymerase chain reaction. And there are some other important parameters for polymerases. One of them is processivity. It's the amount of base pairs synthesized in one sitting, but it's like the one attachment event from polymerase to the DNA template. And the fidelity. Fidelity is the accuracy of replication. So basically the amount of errors that are committed by the polymerase during the synthesis. So you probably want a high processivity and high fidelity on your polymerase, but in some cases, neither of these properties are required. So, and in some cases, you want your polymerase to create some errors in your template for very specific applications. So now that we probably have a target DNA, let's talk about the combination of DNA parts. And to combine some DNA parts, you would want to also use some natural phenomena. And one of these phenomena is a very strange thing that was first found out by a microbiologist. And they found that in some cases, viruses grown on one type of bacteria, they can be restricted from growth in some other types of bacteria. So in this picture on the slide, you can see that there are some strains of bacteria, all kinds of bacteria, and the respective phages that were viruses of bacteria that were grown on this bacteria. So you can see that a phage that was grown on this particular strain, it grows very well on the strain, on its parent strain, basically. Not a parent, but yeah, so it's strain of origin. And it is also true for another strain of E. coli. But in some cases, there are exceptions. And you can see that this particular phage that came from this strain, it cannot really grow very efficiently on this strain of E. coli. And it's a very strange thing because the phage is the same, and the genus of bacteria and the kind of bacteria is basically the same. So what's the deal with this? And it turns out that different strains of bacteria and different bacteria, they can have some very specific systems for recognition of DNA and the cleavage of DNA that is part of its, not immune system, but more like a defense. It's more like a defense mechanism against the foreign DNA. So as a cell, you probably not want for some other DNA, for some foreign DNA with the unknown functions to replicate within you, because maybe it will disrupt your metabolism or I don't know, even overtake all of the processes and eventually lead to the destruction of the cell. So you'd want a mechanism to destroy this foreign DNA, and the restriction modification systems are just the thing for destroying of this foreign DNA, because one of the parts of this restriction modification systems is the enzyme that cleaves DNA, and another part is the enzyme that modifies DNA. They may be combined within the same molecule, but the idea here is that modified DNA is not cleaved, so that it is recognized as the self DNA. And why is that important for genetic engineering? Well, of course, because you can use it to combine some DNA parts from different origins. So you can take one target DNA and splice it to another part of the DNA. So here I have a small questions. So yeah, I remember that I have to interact with the audience. And the question is here. So restriction modification can sometimes be decoupled. So restriction can be provided by one protein and modification by another one. And it was found that aside from wild-type bacteria where restriction is present and modification is present, there are some restriction minus and modification minus that do not have neither. And so there are restriction minus and modification plus, so the only modification. So, but bacteria with the restriction plus and the modification minus were never found. You should probably be able to tell why. Is that so?
Because restriction depends on modification.
No. Oh, it kind of does, but so the restriction, it's more like why bacteria with this particular arrangement do not exist. So restriction is present, but modification isn't.
Excuse me, can be related with methylation?
Oh yeah, it can be. It probably is related.
I think restriction recognize methylated pattern created by modification system, and if modification system was absent and or destroyed, restriction system cannot separate native DNA or viral DNA, for example.
Oh yeah, of course. There's a so of course these restrictases will just cut everything because they cannot be protected by the modification. So that's the answer. And yeah, so it turned out that behind this restriction phenomenon that did not allow some phages to grow on some particular strains of bacteria, there are enzymes that are able to cleave DNA. So they possess the endonuclease activity. And it turned out to be a pretty cool feature for the DNA, for the genetic engineering, because this way you are able to predictably cut DNA and possibly combine them in a predictable manner after this. So I said that it's a cool feature for genetic engineering, but I believe that genetic engineering did not appear before the discovery of restriction enzymes. So basically, it's something that started the field itself. So, and there are actually quite a lot of restriction enzymes. It turned out that not only this particular strain of E. coli possesses this restriction activity, and now people know more than 1,000 systems of restriction modification, and they are divided in several types, and not all of them are equally useful for researchers because some of them they cut in a very unpredictable manner. So they recognize some target, but they cleave far away from it, and in this case, you are not able to predict where exactly it would cut. But one of the types, and it's the second type, it turned out really useful for researchers because it can recognize palindromes, and the methyl acid digestion activities are separated. So you're able to just get the proteins that only cut DNA without modifying it, so that you can ensure that your DNA will be always cleaved and will not be protected at the same time. And also they cut within their recognition sequence or very close to it in a predictable interval, so that you can always rely on getting the very predictable fragments of DNA in every particular case. So yeah, next we are going to be mostly, in this lecture at least, we are mostly going to be talking about the second type of the restriction enzymes. So the way you can refer to them is is provided on the slide, and usually the first part of this nomenclature is omitted because, well, mostly from commercial sources you can get the restriction enzymes, and you're mostly interested in restriction enzymes aside from, well, maybe very specific circumstances. And then the name is comprised of the abbreviated name of the genus and species. In case of this enzyme, it is Escherichia coli, and the enzyme is called the EcoR1. R is the name of the strain, or the abbreviation of the name of the strain, and it usually contains the number of the enzyme in case this particular strain possesses several restriction enzymes, and the number usually reflects the order of discovery. So yeah, the most useful for us are type 2 restriction enzymes because they recognize small palindromic sequences. They produce cuts within the recognition sequence. And the cuts produced by these restriction enzymes can be of two kinds. They can produce the sticky ends and blunt ends. And well, sticky ends are very beneficial for us because you can see that they provide something like a template for hybridization between each other. So in this case, the complementarity between the sticky ends can ensure that DNA fragments are connected in a predictable way. And in this case, you can actually design the construction of DNA molecules from different targets. So yeah, like I mentioned, it is very similar to the hybridization. And these overhangs, they allow specific assembly. And after you cut your DNA with producing sticky ends, and you probably combine DNA from different sources, you want to make it as a whole molecule once again. And to do this, you would once again use the strategy employed by the cell itself. And in this case, you would want to use the enzyme called DNA ligase that requires ATP for reconstruction of sugar phosphate backbone of the DNA strand. And after this, your molecule is basically good to go. You can upload it into a living cell so that you get some kind of a function, or some kind of maybe a protein, or the production of pigment or something like this. So yeah, you can apply the restriction ligation to combine DNA, and so that you can use your target DNA and combine it with the vehicle DNA and upload it to the living cell. So there is some modification to this process to ensure that the vehicle DNA does not ligate on itself, because if you only cut it in one place, your sticky ends that should be compatible with target DNA, they can just ligate onto themselves, and in this case you would get an empty vehicle. So you would probably not want it. You want your payload to be combined with the vehicle DNA. So in this case, you can use a special enzyme, which is called alkaline phosphatase, that removes the 5' phosphate and ensures that the vector does not ligate back after cleavage. So yeah, as you can see on ligation, 5' DNA, 5' phosphate is required for the ligation process, and if the molecule lacks it, well, ligation will not happen. And in this case, it ensures that only the 5' phosphate that comes from the target DNA will be the substrate of a ligation process. Of course, it will end your resultant DNA with well, some breaches in the sugar phosphate backbone, but in most of the cases they are tolerable by the cell, and cell itself will just repair it after the transformation event. So of course there are some other ways to put the target DNA into the vector that ensures the propagation of the target. And one of this is a very simple thing. It's called overhang cloning, and it is based on the fact that the polymerase from a thermostable bacteria Thermus aquaticus, it creates 3' overhangs of the adenine. And if you put the corresponding overhangs of thymine on vector, it can be utilized for the cloning. And yeah, very appropriately, it is named TA cloning. And you can also rely on the process that occurs in cells to produce new combination of DNA. And so the process is called recombination, and it is a process of exchanges of parts between homologous DNA sequences. And recombination can be, so you can recombine molecules on the big stretches of homologous DNA, but you can also use a very specific small parts of the DNA for the recombination. And it's the process that is utilized by the viruses of bacteria, once again, to get into the host genome, to wait for good times so that they can replicate and destroy the host. But if the environment is not very beneficial for them, in any given moment, they can just get, via recombination, they can just get into the host genome and leave there as the part of the genome. So, and this system can be utilized to put some artificial DNA, or I don't know, any kind of target DNA into the vehicle. And of course, the features of this site-specific recombination, it can also be tinkered with. So you can change specificity by modifying the recognition sites, and the fidelity of this process can also be modified so that the recombination can happen with some changed recognition sites. So, and of course, it is used in some cloning systems to put the target DNA into the vector. So yeah, the process that we reviewed in several previous slides, the restriction and ligation, it is akin to the common function of Control X and Control V, because the recognition or restriction you use to cut out some interesting part of the DNA. In this case, it's 2 palindromes, and then you paste it into some other environment, maybe between some control elements that are more suitable to you, maybe in some kind of a vector. So, and the resultant DNA that comes from this process would look like something like this, because so you just cut out some interesting parts and put them into a new environment. You'll get something like a ransom note. And now we are going to be talking about the vehicles, the vectors that are used to make sure that the DNA, the target DNA, is propagated within the living cell. And I don't really know how to think about vectors in case of in linguistic sense. So I thought that it would be something like related to memes that I use to propagate some messages across the multitude of individuals. So you can just, so if you see the funny picture and with the caption, you maybe send it to your friends, and that is the way to make sure that some kind of a meaning is shared across across the community. And yeah, so most of the time, in the basic genetic engineering, vectors that are employed are based on plasmids. And plasmids are very specific extrachromosomal DNA elements that are able to replicate. And well, you might notice that this the number of features of plasmids, the features of plasmids from this definition, it is quite similar to chromosome, and it is actually true that plasmids are not easy to distinguish from chromosomes. And to be honest, there exist some plasmids that are considered just a second chromosome for some bacterial species. And of course, they can vary wildly in size. So there are some very small ones that can maybe carry one function, and there are very huge ones that are basically a second chromosome. And plasmids, they can encode a lot of interesting traits, and they can be passed from bacteria to bacteria because they carry some antibiotic resistance, because they produce antibiotics and kill the rest of the bacteria. So, and the basic idea for plasmid here is that it is a DNA element that wants to reproduce itself, and the things that are encoded by these plasmids are used to gain a competitive edge over the other DNA elements. So if the plasmid encodes some antibiotic, it increases the odds that the cells that carry this plasmid will survive in the environment with this antibiotic. So in this sense, plasmid is some kind of a selfish DNA that just wants to reproduce and wants to stack its odds to achieve this goal. So yeah, here are some examples of plasmids and their encoded traits. And some of these are the antibiotic resistance, some of these are toxin production, and there are some other features within plasmids that are very interesting to us as the researchers and genetic engineers, is that plasmids, well, they want to be replicated, obviously, because that's the point of their existence, probably. And to achieve this end, to achieve this goal, they have to possess a replication origin. So some place where the replication can start. And it turns out that for different plasmids, these replication origins, they may have very different mechanisms. And so plasmids can use a wide array of proteins, of DNA sequences that help them to replicate it in the host cell. And why is it important to us? It turns out that the plasmids with different replication origins can cohabitate within the cell, and the plasmids with the same replication origin will compete for their existence within the cell. And so, yeah, well, plasmids with the same replication origin, eventually one of them will overtake the cell, and the other one will disappear. So, and it is the basis for the plasmid incompatibility. That is a very useful trait for genetic engineering, because if you plan your genetic engineering experiments, if you want to use some plasmid as a vector, you need to take into account its incompatibility group, so that you do not put two plasmids with the target DNA into the same cell with the same incompatibility group. And the DNA plasmid replication origins also correspond to the copy number of a plasmid. It is defined by the origin of replications, and some plasmids are very high copy number. And in this case, well, there are several hundreds of these plasmids in the cell. And some of the plasmids are very low copy number. So there are some plasmids that only exist in a couple. There are only a couple of these plasmids at any given moment of the cell. And in this case, you would want to make sure that the plasmid that has a very low copy number, it somehow ensures that it is passed down the generations. So, and to this end, some of the plasmid, they possess some mechanisms for partitioning into daughter cells. So for high copy numbers, you may see it's not a problem, because if there are several hundred of them during the division, some of them will be randomly assigned to two daughter cells. But it's not the case for the very low copy number plasmids. And in this case, some of the plasmids, they employ systems very much alike to the partitioning of chromosomes in the eukaryotes, because because they have some kind of a semblance of centromere, and they use fibrilla to push out two copies of the plasmids into two daughter cells, so that both of them will get their share of the plasmid DNA. So yeah, here's the incompatibility feature that I already talked about, and you can see from this table that there are some groups of incompatibility, and some of them several groups of incompatibility, and of course you cannot put the plasmids from the same incompatibility group into the same cell. And so there are several something like 30 incompatibility groups in E. coli. And the good thing about incompatibility that can be engineered. So if you alter the way that the plasmid replicates, somehow tinker with its replication origin, you can also change its incompatibility group. And in this case, you'll be able to use two plasmids previously incompatible at the same time. So another important feature about the plasmids is that the competitive edge that allows them to survive and to be propagated in living cells. And in natural circumstances, it may be something like toxin antitoxin systems that are employed by R1 plasmids of E. coli, for instance. And in this case, the idea here is that plasmid can make sure that if the cell loses the plasmid DNA for some reason, the cell will swiftly die after this, because it produces two kinds of the RNA. One of them encodes the toxin protein, and this mRNA is quite stable, and the antidote for this toxin, in a sense, because it's an antisense mRNA, it degrades quite rapidly, but it is produced constantly if the plasmid is present. So that if you have the plasmid still in the cell, the antidote will be still produced, and the mRNA that encodes toxin will not be translated, which prevents the death of the cell. But if you lose the plasmid, of course, the short-lived mRNA will disappear, and at some point the toxin will produce that will kill the host cell. In case of the genetic engineering and the human application of this selection systems by humans, you would maybe want to use some other features, because killing the cells that do not carry the interesting DNA is a good thing, but in some cases you would want to also counter select the cells so that cells that do not have some kind of modifications, they can also be picked up. So in some cases you would want to utilize some different strategy for the selection. And so one of these selection types is the utilization of oxotrophy. And the oxotrophy is the inability to synthesize a particular compound that is required for the organism's existence. And so mutants that are unable to produce some kind of compound are usually denoted by the name of this compound, the abbreviation of it, with the minus. So if you are unable to produce the vitamin B, for instance, the corresponding strain of bacteria will be called a B minus. And the opposite trait, it means that you can produce everything that you need, is called the prototrophy. So you can see that prototrophs that can produce everything they need, they can grow on the minimal media without adding some food supplements. And the bacteria that are oxotrophic for something, in this case for arginine, they are not able to grow without the addition of the arginine. So you can see that on minimal media they die, and if you add this amino acid, they can divide and replicate, and everything will be all right for them. So another question for the audience is are humans oxotrophic for anything, and are plants oxotrophic for anything?
Humans for vitamin C, I think. And also for those 10 essential amino acids.
Oh, yeah. So yeah, for humans it's true, and for plants, are they oxotrophic for anything?
Well, I mean, they do need the minerals. So.
Oh yeah, I think they cannot exist without the supply of at least something, because you need to consume something. But they can produce most of the things. So yeah, at least we are oxotrophic for more stuff than plants in this case. And yeah, of course, another very famous example for human oxotrophy is the vitamin C oxotrophy, because with some high primates and guinea pigs, they, for different reasons, cannot produce the vitamin C. And in our case, the inability to produce the vitamin C resulted in the scurvy in the ancient mariners. And in case of guinea pigs, I don't really know. Maybe they do not undertake this kind of adventures, but yeah, they are also not able to produce the vitamin C.
I think also B12 vitamin.
Oh yeah, a lot of stuff. I just wanted to put this example for the guinea pig picture. So another strategy to select for organisms with a modification is to employ the antibiotics. And as I mentioned, a lot of plasmids, they carry antibiotics resistance genes. And well, antibiotics is basically a weapon of warfare between different microorganisms. And as a result of this arms race, there are a lot of antibiotics that target just about any essential process within the cell. So a lot of them are targeting the protein synthesis, some of them target the transcription, some of them target well, the synthesis of cell wall. And well, you can use a lot of this stuff on both prokaryotes and eukaryotes. So a lot of the things are, well, they're equally deadly for all of the organisms, because, well, some of the template processes are very similar. And yeah, so that to utilize it as the selection marker, you would want to put into your vehicle some kind of a antibiotic resistant genes, and some plasmids already carry them, of course.
So there are a lot of ways you can achieve the antibiotic resistance as a trait, and some of them are not all of them are equally useful for genetic engineering, of course, because some of them may be well, they're not so easily put into plasmid. For instance, if you modify the target of the antibiotic, like the ribosomal RNA or some ribosomal proteins, it is not so easily put into the plasmid. But some of the other stuff, like an activation of the antibiotic or pumping out of the antibiotics, it can easily be put into the plasmid to use a selection marker. Of course, the impermeability, the reinforcement of the cell wall is also not such an easy trait to put into the plasmid. So yeah, and talking about antibiotics, it turns out that, well, you probably know this, but antibiotic resistance that is used as a selection marker is also a global threat to the worldwide health, because it is put number 5 on the list of the health threats last year. And well, there are some strains that have resistance for just about anything, and it's not a good thing, because some of these bacteria are quite dangerous, and if you are not able to treat them with the antibiotics, then your chances of surviving them are probably really low, and if they spread further, they may be quite dangerous for us, the humanity. So here on the slide is one of the first plasmids that was used for genetic engineering, and you can see on the picture on the right that it has, well, all of the stuff that is required from the vehicle in genetic engineering. So it has something to replicate, so the replication origin. It has the actually it has two antibiotic resistance genes, because one of them is used as the site for putting the target DNA, and in this case it is used as the selection marker that shows that if you lose this antibiotics resistance, that means that something was injected in this particular site. So you can use some of the restriction sites within this gene to put some target DNA here, and if you lose the resistance to tetracycline, it probably means that your target DNA was incorporated into the vehicle. So the way you clone with this plasmid is that you put your plasmid with after the restriction ligation. You hopefully it was the successful event, but maybe in some cases the intact plasmid remained in the cell. So you put it on the growth medium that has one of the antibiotics, ampicillin in this case. And if they grow, it means that at least they have some form of the plasmid, modified or not, we don't really know, but we can't tell right now. And for the next part, you need to replicate this plate. So you maybe take a piece of paper, you put it here, and then you print it on the next medium that contains tetracycline. And if cells survive on the tetracycline, it means that the plasmid was not modified in this site, and it means that, well, no modification occurred, or probably, well, target DNA was not incorporated here. So, and if they do not grow on the tetracycline, you take a note that this colonia of bacteria, they did not grow, and so you can assume that they were modified with the target DNA. In some cases, people do not use the singular sites of the restriction, and they combine them into very long stretches of DNA, artificially engineered. And these stretches of DNA are called polylinkers, or multiple cloning sites, and they contain a lot of overlapping the restriction enzyme recognition sites. And it is used for researchers' convenience, because you're not required to buy some specific enzyme, for instance, to perform your cloning experiment, and you can use maybe all of this at the same time. You can also use two of them so that you can ensure the directionality of your insert. And yeah, so you have some freedom of choice in this case, and use a lot of different restriction enzymes. So there are some other strategies for counter selection, of course, and not all of them require the plating of first colonies onto the second growth medium. And one of such systems utilizes the blue-white selection. And the idea here is that, well, the insert of the target DNA disrupts another gene, not an antibiotic resistance gene, but a gene that can produce the coloring of the colony. And in this case, it's a beta-galactosidase gene. And in case you disrupt it, it loses its enzymatic activity and is not able to produce the blue coloring of a specific substrate. So there is the substrate, which is called XGAL, and in itself it is colorless. But if you subject it to beta-galactosidase, it will produce the dark blue compound, and it is easily detectable on the plate. And in this case, you can see that blue colonies, of course, do not contain the modified genes, so they probably do not have any insert. And you should not use them in your downstream analysis. So probably they do not have any foreign DNA insertion. So a little bit about the bioinformatics applications here. So you might have noticed that people want to get a lot of people want to use a lot of restriction enzymes. They want to use a lot of maybe selection systems, and for this you may want to somehow mine the available resources of sequences, so some metagenomic data sets or genomic data sets, or maybe sequence something de novo and then subject it to finding some new plasmids and finding some new traits to be selected for. And you can perform it with the short read sequencing, of course, and in this case you may use some specialized tools that are able to reconstitute plasmids from short NGS reads, like plasmid spades, not advertisement in any form. And also in the recent years, with the rise of nanopore sequencing, you can also use the long reads to assemble plasmids. And in this case, this particular example is for finding plasmids in some pathogenic bacteria. So it's not exactly for genetic engineering, but rather for finding the resistance of clinical strains of some bacteria. And so the thing about them is that if you detect it in your patients, you would probably want to know what it is resistant for, so that you can choose some other antibiotics to kill it within the patient. And the traditional strategy would be to use the media with the different antibiotics plated on them. And it may take a lot of time, especially if bacteria do not have a very high replication rate, so which is probably the case for just about everything aside for Escherichia coli. And in case of sequencing, you can achieve the same goal, but much faster, because using long reads, you can just take probably in some cases a very small amount of reads, like maybe 1,000, to get the complete coverage of the plasmid. And from this complete coverage, you can deduce the antibiotic resistant of this particular plasmid and the bacteria that you're sequenced. And yeah, here on the second graph, you can see that for different types of bacteria, well, a very small amount of time is required to achieve the operable coverage, so that you can deduce the antibiotic resistance. So it is probably much faster than the traditional strategy, so that you can get to the treating much faster. All right, and the last part about the basic genetic engineering is the host cells. So I was talking about the creating the recombinant DNA, but in the end, you would want to use the replication machinery and the translation and transcription. And of course, you would want to use some kind of a chassis for this, the host cell that will carry out all the necessary stuff to produce the product from the recombinant DNA. And the host of choice for most of the basic genetic engineering is of course, E. coli, and it is a gram-negative bacterium, and we know quite a lot about it because it was sequenced a lot of times, so we know a lot about the different variations of this organism. And the best feature about it is that it replicates really, really fast. So there are some other cool features, but this one is very convenient for genetic engineering, is that you can replicate any kind of DNA in a very short time. And the interesting thing about it is that, well, the notion of species in bacteria is very strange, in the sense that the thing that we can call E. coli can differ very wildly. And of course, the bacteria that caused the enterohemorrhagic disease in people several years ago are not the same as the bacteria that we are using in the everyday lab environment. So of course, there are different strains that well, may contain a very diverse set of genes. So strain is basically a frozen a photo, in a sense, of a bacterial species, because you take it from a very specified location, or bacterial virus, of course, and you can take it from a very specific location at a specific time, and it probably has a clonal origin, in a sense that it is all of these cells from the same strain, they probably come from the single progenitor. And this feature allows strains to be very genetically universal and very genetically uniform. And well, you can see that strains can vary very seriously between each other, because, well, within the single bacterial species, there are only there is some proportion of core genes that are required to be contained within the species, but there are quite a lot of accessory genes also that can, well, do a lot of stuff, and they can differ very, very wildly between strains, causing some of them to be pathogenic and some of them to be very benign and applicable in lab environments. So the way you characterize strain in the practical application is that you'd want to know the genotype of the strain. So, and in this genotype, usually features, usually reflected are features that are useful for your genetic engineering experiments. Like for instance, in this case, one of the most popular E. coli strains, DH5 alpha, is described, and you can see that it lacks homologous recombination, which allows you to make sure that the foreign DNA that you introduce there is more stable, so it is not, well, jumbled by the recombination systems within the cell. If you by any chance have stretches of homology between the foreign DNA and the chromosome. Also, it allows for this strain, one particular mutation in endonuclease. It allows for lower degradation, and it ensures that plasmid can actually survive within the cell. And it also carries a mutation that allows for blue-white screening of recombinant cells, because if this cell has a chromosomal copy of beta-galactosidase, of course, all of the colonies will be blue, because well, the cell has a functioning enzyme, and in this case, the target targeted insertion will not disrupt anything. So yeah. So after you select a proper strain for your particular application, you would want to put the foreign DNA into the cell. And this process is called the transformation for bacterial and plant cells. And for animal cells, it is called transfection, because transformation for animal cells is something else. So basic idea behind transformation is that it is an uptake and an incorporation of external genetic material, and the ability to do this is called competence. And it turns out that a lot of bacteria are able to take the foreign genetic material, because it's a very cool feature to find useful DNA fragments in the environment and use them to your ends. So how it was found is that another way of genetic of transferring of genetic material between bacteria is that in some early experiments on bacterial crosses, people sometimes found out that if they crossed the oxotrophic mutants, so one of the mutants is not able to produce some of the compounds, another mutant is not able to produce another compounds. I believe most of these are amino acids, and this is a vitamin B or biotin, I think. So sometimes crosses resulted in the prototrophy of descendants. So some of the crosses were so some of the strains of the bacteria were compatible, meaning that they can actually cross between each other and produce these prototrophic descendants, and some of the strains were not able to do this. And of course, the underlying of this, so underpinning of this, was another process of genetic material exchange between bacteria. So here I have a strange question for the audience. Is that so you remember that, well, if you cross the F plus and F minus, they can exchange genetic material. And during one of the experiments, it turned out that if you try to cross these strains of bacteria, F plus and F minus, with these phenotypes, they should, well, theoretically, they should produce the prototrophic descendants, prototrophic progeny. But if you shake it vigorously, the crossing was not productive. And maybe any ideas on why did this happen?
It disturbed the pili that do.
Well, yeah, it was, yeah, of course. So it distracted the conjugation pili that are required for the exchange of the genetic material. It was also very strange to read it in the paper, that people did not really know it at the time, and they have this strange result. So yeah, that's another way to exchange the genetic material. But for transformation, well, some of the cells, they possess the natural competence, so that they are able to uptake the DNA from the environment as it is. And for this, they also have some kind of pili to make sure that they contact with the DNA, and then they have the channels that are able to put single-strand DNA inside the cell, and later this single strand may be incorporated, may be recombined into the chromosome, and maybe if this outside DNA was useful, you can maybe obtain a trait of antibiotic resistance or something else. So, and it is not always true that bacteria are competent, because, well, some of the bacteria, they lack this natural competence trait. So in this case, you can
actually induce it by different agents, and it can be induced chemically by the application of salts and the heat shock, so that the membrane of the cell is disrupted enough so that the DNA can enter the cell, or it can be disrupted by the electric shock provided to the culture of bacteria, and well, basically do the same end. So you end up getting the plasmid inside the cell. And so that concludes the overall structure of the basic genetic engineering experiment, is that, well, you take a target DNA, combine it with vector DNA, possibly a plasmid, and you introduce it into the host cell by the transformation, and hopefully your cell will produce some kind of a protein that was encoded by the target DNA. So, and all of the process that we reviewed, it is called the molecular cloning, and it can be compared to the, well, selecting everything, in this case the vector with the insert, copying it and pasting it a lot of times, because this DNA construct can be replicated multiple times in a lot of generation of host cells, so you can get a huge number of copies of it. So yeah, another question for the audience is that you may want to use the restriction enzyme coding sequence as the target DNA. So you may want to use it as the as a target DNA. So you may want to produce the restriction enzymes to sell it later. So you can maybe put it into some plasmid, and you would want to express it in some host. And what would you think would be the obstacles in this case?
Maybe some silencing mechanism. So if you have restriction enzyme with a certain target, we can conclude that some DNA fragments will be restricted if they have the restriction site.
Now, you mean some other genes in the cell, or?
Yeah, maybe some other vectors.
Yeah, okay. So in this case, I put the restriction enzyme coding sequence and the maybe modification enzyme coding sequence, and still it may not work for some reason. So yeah, you can use the modification enzyme for protection, but may it also, is there a reason for it to not work in certain circumstances?
Do you mean if you use the one vector with restriction enzyme sequence and some other gene? Or...
No, I mean so you would want to produce the restriction enzyme and sell it, because some of them are quite expensive. So it's a valid strategy to use some kind of bacteria to grow them, to put a restriction enzyme coding sequence into them, and to grow a lot of restriction enzyme protein from them and to sell it later. And one of the obstacles may be that this restriction enzyme will cut host DNA, but you can maybe circumvent this problem by putting the modification protein.
I think the deal in here, it's right promoters. We should choose right promoter. In our case, it's something promoter from plants, and replace in vector.
Well, assuming that you use the right promoter, for instance, you want to replicate it in bacteria, and you use the corresponding bacterial promoter. So the protein is actually produced.
You should have a zinc finger domain to produce the proteins.
Why?
Because if it will be just a restriction enzyme, it will be just 4 or 5 nucleotides.
Wait. Restriction enzymes are quite large. So
Yeah.
6 nucleotides is recognition site.
Mm-hmm.
So restriction enzyme is a normal protein, basically. So the coding sequence will be, I don't know, maybe 1,500 nucleotides, depending on the enzyme. So it's not going to be short, for sure.
It depends on origin of plasmid or vector.
Assuming that we use the normal origin of replication, so the suitable one. So it actually works within the cell.
Okay, we made already. No, we have made already transformation or not?
Yeah. So everything is working as it should, and you have a plasmid inside the cell. You put the restriction enzyme and the modification enzyme, and so it can readily reproduce within the host.
And everything is
Our vector can able to insert in genome, plant genome, or not?
No.
Because it's
maybe because polymerase activity.
What do you mean by this?
Okay, so we have a transferred restriction enzyme in cell, and we want to produce the protein, and we want to get the gene expression.
Yeah, the question more specifically is that, so there is a real life situation that some of the restriction enzymes cannot be produced in foreign organisms. Well, at least there are problems in doing so, because, well, some of the restriction enzymes as a result are very expensive, right? So they can only be produced in their specific host bacteria, and they cannot be readily transferred to E. coli, for instance. And what may be the reason for this? So,
Is it the temperature?
well, temperature may be the problem, yeah.
maybe it's depends on cell division. For example, if our plasmid do not integrate in genome, cell divisions of plants make decreasing of concentration of vectors, and finally we have some cell from plants which do not have which have not plasmids.
Well, yeah, in practice, host organisms, they try to lose the plasmid because it provides a metabolic strain on them. Because if some other genetic element requires you to produce some more proteins to transcribe it, it is hard for the cell, and they tend to lose plasmids. So yeah, all right.
I guess also some restriction enzymes connect with methyltransferases. So if we want to insert this type of restriction enzyme, we should also insert the modification enzyme, because the activities are connected with each other. And I guess also some restriction enzymes can require specific chaperones for 3D structure building, and some bacteria can have the appropriate chaperones.
Oh yeah, all right. It sounds great, because I don't really know the exact answer for this question, because there may be some valid points, because if you put modification enzyme, for instance, it can produce some unnatural methylation that will lead to the cell death. or yeah, maybe the enzymes require some specific assembly that is not available in the new host cell, something like this. So yeah, all right. So that's the end of the first part of today's lectures, and here's the recommended reading, but I'm pretty sure that you don't really need to read all of these books. You can just, well, maybe use them as a reference, because I will use some. You probably already know a lot about genetic engineering, and I will use it without the explanation during the course, at least some parts of the course. So maybe just refer to them if needed. So from now, I think we should have a small break so that we can go to the second lecture, maybe 15 minutes, or is that all right with you? Hello?
Yeah, okay.
All right, so see you in 15 minutes. So the second part today is going to be about the genome editing, and it's going to be slightly better because it is at least modern in the sense that it's a very vibrant and new field in biotechnology and biology. So yeah, the outline of the lecture here is well, what do we do about the genome editing, and then we'll review some systems for genome editing and the practical applications of them. So the basic idea here is that for any genetic modifications, you probably need some kind of a material. So modifications means that, well, there was something like this initially, and either through carefully picked the breeding programs, like in case of dogs, or in case of plants, you get your new traits by the induced mutagenesis. You end up with a new organism with a beneficial traits. In case of corn, it has more seeds, and they are bigger than the in wild counterparts. And in case of dogs, maybe they are more cute or something like this. So in either case, you would want to get some diversity first, and then you want to somehow select for it. And genome editing is something that tries to subvert this, because in this case, you produce targeted changes. So you do not need to wait for new interesting traits to spontaneously occur or occur through the chemical inducing. And in this case, you want to target very specific parts of genetic material and make sure that it produces a specific new phenotype. Like maybe you want to target a locus that changes the eye color, and in this case, it will be a very precise modification. So, and for these targeted genetic changes, of course, as the name implies, you would want two things: targeting and changes. And good thing that targeting part of the description is already provided by the nature itself, and it already exists within cells. So one of the obvious things that does the targeting within cells are transcription factors. And of course, they serve to transcribe some specific subsets of genes within a genome. And to do this, they recognize and bind DNA. So they do not bind everywhere, and they hopefully bind very specific subsets that will produce a very particular transcription program. And this in normal organisms, they regulate the functioning of genes so that they repress or activate something, and it leads to, I don't know, in the aberrant cases, it may lead to the substitution of antennae with legs, and so on and so forth. So and also to main normal phenotypes as well. Of course, transcription factors have to somehow zero in onto targets, into their targets, into some specific DNA sequences. the recognition sites. And well, it turns out that the transcription factors, they have a set of rules for this. So they do not just bind, well, DNA in general, they actually have preferences for within the DNA molecule to interact with some preferential sites. And mostly transcription factors interact with the major groove of the DNA double helix. And in this major groove, they recognize a specific set of donors and acceptors of hydrogen, for instance, or in some cases methyl groups, and it allows them to recognize. So they do not need to gain access to the nucleotide bases themselves to recognize the sequence, like in DNA hybridizations. It suffices them to just approach the DNA casually through the major groove and to sense these groups within the major groove. So some transcription factors are also able to recognize the minor group, but I guess it's whatever. So in any case, transcription factors, they can recognize DNA through this interaction with the DNA molecule. And in some cases, these rules for the transcription factors recognition are actually quite understandable for us. And in this case, we can, if they are reproducible, understandable, you can maybe employ them to reproduce the binding activity in artificial proteins. So one of such cases, the zinc fingers transcription factors that were mentioned previously. And well, it's one of the transcription factor classes. They are usually classified by the DNA binding elements. And well, parts of this DNA binding domain, in this case, they interact with the zinc ion. And it turns out that, well, zinc fingers, well, individual entities within this DNA binding domain, they recognize very specific subsequences within the well, transcription factor recognition site. Like, for instance, this particular zinc fingers, it recognizes two of these nucleotide pairs. I don't really know the exact rules, but it turns out that they are, in fact, you can produce a catalog of them. And in this case, if you have a compendium of all these recognition rules, you can place several interconnected zinc fingers and create a protein that is able to bind to a specific sequence. So if you stack a lot of these zinc fingers, you end up with a zinc finger array, and this array will hopefully bind some specific DNA sequence, and it's not going to be a very short one, so that you can ensure that it is actually specific in its binding. So now that we at least have one mechanism of the targeting, so if we stack a lot of zinc fingers, so you can maybe recognize a very specific sequence within the genome, you want to get to the editing part. So the editing part, of course, can be done with a lot of things, but for now, we assume that we only know about the restriction enzymes, and it turns out that it is sufficient knowledge for our target editing, because the most basic option for mutation, in case when we are working with the genome DNA, is the cutting, because the way the cell treats the cut in the DNA, especially a double-stranded cut, is that sometimes it introduces errors in the cut site, and it will look like a disruption of the maybe coding sequence if it's the protein coding DNA and something like this. So maybe it will turn from functional state to a dysfunctional state, and you'll get a mutation in a very specific site that was determined by the transcription factor recognition. And combination of these ideas, the zinc finger arrays and the nuclease activity of restriction enzymes, it turned out into the idea of zinc finger nucleases, and I think it's one of the first genome editing technologies, and it is quite simple. It's a hybrid protein with DNA binding domain and the nuclease domain of a very specific restriction enzyme. And good feature about this restriction enzyme is that, well, first of all, the cleavage activity can be easily separated from the DNA binding activity. It is not always true for restriction enzymes, but in this case, the cleavage domain can be cut out from the DNA targeting domain. And it can be, well, spliced with the zinc finger array, and so that you get a targeted binding and the cutting. And also another cool feature is that it requires dimerization for cutting. So it produces a double-stranded break in the DNA sequence, and by dimerization also ensures that you get a longer stretch of DNA as your target, so that you can achieve even higher specificity. So of course, well, maybe a sequence of 10 nucleotides will occur here and there in the genome, but if you target for 20 nucleotides, it will not be so widespread in the genome. So yeah, here's the question: why cutting leads to a mutation? And it turns out that the double strand break is a very bad thing for a cell, because it may turn into a cell death, because, well, cells usually do not tolerate this kind of damage when they're checking for the readiness for the division. And if they sense double strand break, it may be better for the cell from point of view of the guardian mechanism of division to just abort the existence of this cell, so that it does not produce some kind of cancer cell that can jeopardize the existence of the entire organism. And of course, cell does not just die immediately. It tries to repair this kind of very dangerous damage. And there are, well, at least two major mechanisms. There are more. There are some intermediate stuff here between them. And one of the mechanisms is the non-homologous end joining, and another one is the homologous recombination. And the culprit for the mutation interaction here is the non-homologous end joining, because it means that if the double strand is repaired by this mechanism, it will probably introduce some kind of mutation, because it inserts or deletes some random number of nucleotides. In case of homologous recombination, another surviving strand that does not have a double strand break. So usually in the diploid organs, you have two chromosomes, and if one of them is broken, the other one is going to be used as the template for the repair. And it results in the precise gene modification, meaning that, well, there are no mutations. So it's probably bad if you want to introduce a mutation, but it is good if you want to put, well, some huge genetic fragment here within the same locus. So in this case, the homologous recombination is your friend. And yeah, usually the relative frequency of this mechanism, it depends on the organisms. There are some organisms that use homologous recombination a lot, but usually, especially in higher eukaryotes and in humans, non-homologous end joining, it happens more often. And yeah, it turns out that it's pretty all right for us, because non-homologous end joining can still is good enough for a lot of genome editing applications. But in some cases, in case you want to introduce something with the homologous recombination, you may want to somehow, well, alter the relative frequency of these two mechanisms. And there are ways to do this. So one way, one avenue for this would be to overproduce the genes that participate in the homologous recombination, and, well, another way is to suppress the homologous end joining. And in some cases, also you can try to introduce more homologous recombination by also increasing the stretch of homology. So if you want to introduce some gene, you want to have it flanked with the homology arms, so that it is inserted in some particular place in the genome. So if you want to increase the homologous recombination, you may want to increase the length of these homology arms, for instance. But there are a lot of ways to do this. So you can maybe suppress non-homologous unjoining, maybe somehow overproduce the homologous recombination parts. And so some of these approaches actually work and increase tangibly the amount of the products that were produced in the homologous recombinations. All right, so now for the examples of the zinc finger nucleases and examples of the usage. And one of these examples is connected with the HIV, human immunodeficiency virus. And the idea here is that, well, as we know it, it targets the immune cells within our bodies. And to do so, it has to bind to its receptor. And one of the, it also has to bind to one of the co-receptors on the surface of the immune cell. And one of these co-receptors is called CCR5. And so it is required for the infection of the cell by the HIV. And it turns out that some of the Earth human population, especially this mutation is widespread in Northern Europe, some of the people are well, resistant to HIV. So to achieve this resistance, well, maybe they do not achieve anything, but they have a very specific deletion in this CCR5 gene that ensures that their cells are not infected by the HIV, at least the HIV strains that are that require CCR5 infection. So it may be very cool to introduce this kind of deletion to, well, maybe some people that already are infected by the HIV, and in this case, maybe virus will not be able to infect the new cells. And so that's a great idea for genetic engineering efforts, for genome editing in particular, because it has a very specific goal. So you just need to delete this part of the gene, and that's it. So you probably win if you have a CCR5 tropic strain of the HIV. So, and what's even better about the situation is that there are actually examples of people that were cured of HIV in this way. So the first person that was cured this way he's known as the Berlin patient, and his identity is, well, already known. Timothy Ray Brown is his name. And he was quite a lucky person because on top of having AIDS, so he was infected with HIV, he also had an acute myeloid leukemia. So AIDS and cancer at the same time. And it allowed him to, well, first of all, it allowed him to undergo treatment from cancer and AIDS at the same time. And so he was treated from cancer by irradiation. So his leukocytes were basically destroyed. And to provide the substitute for them, he was given the donor bone marrow from a person that has this HIV resistant mutation. So from a person with the CCR5 delta 32. And it turned out that with this bone marrow, he became unsusceptible to the HIV, and as of now, he is, well, functionally cured from the HIV. And what's even more, it's not a fluke, because this year there was another patient. He is called a London patient, and his name is also known. I don't really remember the last name is Castillejo. I don't really remember the first name, but he had the same combination basically. So he was infected by the HIV, and he had the leukemia at the same time. And the same treatment basically resulted in the fact that now people are tiptoeing around the term cured, because well, he's still being tested. It's very recent news, but basically it shows that this approach may work. So if you want to cure the HIV infection, so And it's not a coincidence. Maybe it's not a happenstance that helped the Berlin patient be cured. So this approach was actually used in some experiments, in this case on mice, and it turned out that virus titer was quite reduced, and in the mice that undergone this kind of modification, so they used the zinc finger nuclease to introduce this delta 32 mutation, and well, CD4 plus cells also survived, so they did not die because of the virus infection. And several weeks after the inoculation, the load was almost undetectable, even in the pools that I usually checked for the HIV, so in the intestine. So in mice, at least it works if you use the genome modification for the HIV treatment. And because this approach may actually work, there are some biotechnological companies that use this zinc finger nucleases to achieve some medical goals, to treat some diseases. I think an interesting case. So there is a company that's called Sangamo, and it has a lot of treatments in the pipeline right now. Maybe even more this year. I haven't checked, but an interesting case of treatment of beta thalassemia here is provided on the slide, and they of course used once again the zinc finger nucleases. And in this case, it is interesting in the sense that they do not alter the protein sequence and the protein function itself, because, well, in case of beta thalassemia, as you may know, the problem is that people have some kind of defects in the beta hemoglobin, and well, it's really a bad thing because, well, in this case, erythrocytes cannot normally carry enough oxygen for the person to normally exist. And yeah, so you may think that the treatment would be to rectify the sequence of the beta globin, but it's not the case here, because people are even more kind that design this treatment, because they utilize the fact that people have a lot of different globines in the same locus. And the globines are switched when we are transferring from our fetal stage of life to our postnatal stage of life. So one of the globins is substituted by another. So when we are after birth, the beta globin overtakes the function of the gamma globin. And it turns out that you can functionally treat this disease if you just turn on the fetal gamma globin. So you just need to tinker with the control elements of this locus so that this gamma globin is expressed once again, and it can achieve the same goal as the given the functional copy of the beta globin. Maybe it will function slightly differently, I don't know, because I think that hemoglobins, they differ in the oxygen binding activity, but I think it's an interesting case and would be great if it actually worked. I'm not sure what stage of the clinical studies it is in right now. So talking about the advantages and disadvantages of zinc finger nucleases is that, well, at least about downsides of it. So advantages are obvious because you can use it to the genome editing, and I don't know, it just works. So you have a lot of proven things that are true about the zinc finger nucleases. But the downsides are that zinc finger nucleases are quite hard to engineer because it requires that you assemble it from the protein domains, and so that if you want to switch target at some point of your experiment, you would need to combine a whole new array of zinc fingers. And also, it was true, at least at some point, for zinc finger nucleases, that they give a very high off-target effect. So maybe the rules for the recognition for zinc fingers are not so robust, and so that if you produce some kind of zinc finger array, it may actually produce changes in some loci that were not originally intended for the edition. And it is quite bad because, well, maybe in some cases these changes will result in the lethal modifications that, well, are not compatible with life after this well, genome editing event. Hopefully, the very same company that relies on the zinc finger nucleases, they try to counteract the downsides of the zinc finger nucleases, and they say that they have a zinc finger nuclease that have higher precision, higher efficiency, and well, less off-target activity. So we'll see what the future holds for this, but probably. So I don't think the sequences for this modified zinc finger nucleases are published, but hopefully they will work better than the ones that were, well, previously used in the genome editing. So yeah, that concludes our talk about the zinc finger nucleases. So we talked that there is at least zinc fingers were one of the sources of the specific interaction between the target DNA and some kind of a modifying protein. But there are some other sources of these types of interaction, and one of them comes from the pathogen-host interaction between plants and bacteria. So, and the good thing about it is that, well, hosts and parasites, they have a constant arms race. And this type of interaction, it requires that both of them should have some kind of a very specific tools to, well, counteract each other. And in case of the interaction between Xanthomonas and plant cells, it resulted in the, well, origin of proteins that are called transcription actuator-like effectors, or in short, TAIL. And bacteria, they use them to counteract plant immunity. So bacteria, when they interact with plants, they introduce, through the secretion systems, they introduce some kind of protein, some types of proteins inside the plant cell, and they activate genes that are beneficial to bacterial infection. And, well, of course, plants do not really want bacteria to activate these types of genes, so that they change the promoter sequence for this type of genes. So it's the way they try to elude this unwanted transcription activation. But in turn, bacteria can also change their effectors once again, so that they are compatible with the new promoter structure, so on and so forth. So this is like a cat and mouse game between them, and which is why, well, tail effectors, they have a very specific structure. And so these tail effectors, so yeah, that's basically what I said previously. So they have a very specific structure, so that they are composed of repeats. And these repeats, each of them, they are very small amino acid repeats. They have a very strict specificity to some of the nucleotides in the correspondent DNA sequence. And these rules can once again be put into a catalog, and you can use these rules to assemble an array of these tail repeats of any given specificity. And yeah, the way tail binds to DNA is that it interacts through the major groove, and the flexibility is achieved through this modular structure. So even if you change one nucleotide here in the promoter, it, well, probably does not matter too much to the bacteria, because they can switch the repeat here and still be specific to this promoter. So here's what tail repeat look like, and they are small amino acid repeats, and the recognition function is performed by this pair of amino acids approximately in the middle of this repeat. So if you if you investigate it thoroughly and find all of these specificities between these deamino acids and the nucleotides, you can basically assemble an array with any given specificity. So people, of course, did this, and they end up with a tail code. So now we can, well, much like the zinc finger arrays, we can assemble an array of tail repeats so that it targets a very specific part of the genome. So, of course, like in the previous case, you would want some kind of a nuclease activities. And in this case, once again, the restriction enzyme FOC1 can provide it for you. So you once again use this part of this enzyme that cuts the DNA. And once again, it is very cool that it needs to be dimerized to cut anything at all. And yeah, in this case, you can get a high specificity, so higher than in case of a non-dimer cutting. So of course, it was discovered slightly later than zinc finger arrays, but there are, well, multiple applications of this tail nucleases, TALEN, and they are used, well, in both academia, and I think that there were at least some trials to use them in clinic. So in this case, it's a purely academic research. It was used to study Gibberellin signaling in plants. So one mutation was introduced through the disruption of a particular gene with the TALEN system. So downsides of tail nucleases it is once again hard to engineer, because if you want to swap sites, recognition sites for your experiment, you once again need to come up with basically a new protein. So you need to start from square one every time you want to use a different target sequence in the genome. So yeah, off target is slightly better for TALEN because they are even considered quite accurate compared to our next genome editing system that we're going to review last. So, and the last, of course, the most famous of them, genome editing system, comes from the bacterial immunity. And so, of course, bacteria need to fight the foreign DNA sequences. And one way to do this is to use the restriction enzymes. And another way is to use, well, actually, what we are going to be reviewing is also considered now a restriction enzyme. So we basically use the restriction enzyme for this. So the idea here is that, well, the concept of immunity means that you are probably able to remember the past infections so that you can fight them more effectively if you meet them once again. So, and to do this, the bacteria, they use CRISPR-Cas systems, and CRISPR stands for clustered regularly interspaced short palindromic repeats. And so this is how they were discovered. So basically it was found that in bacterial genomes, there exist some strange clusters of repeats. And well, and as the name says, the palindromic repeats were considered maybe a more prominent feature of these clusters at the time. But to be honest, the most important part about this are actually spacers. So repeats are features that, well, separate the spacers, but the functional part here is what lies between these repeats. So the mechanism here is that the viral DNA at some point gets into the cell, and maybe in some circumstances, Cas proteins, they cleave the viral DNA and insert a part of it into the CRISPR array. So later, these CRISPR arrays are transcribed, processed, and used to recognize and cleave this viral DNA. And well, to do this, they may use a lot of different proteins. The most basic part, well, there are a lot of CRISPR-Cas systems, but the basic processes that are required for this CRISPR-Cas functioning are protein that actually cleaves the DNA, so they can counteract the foreign DNA, and some proteins that are processing the CRISPR array, that what is transcribed from CRISPR array. So first of all, you transcribe it, then you cut it into small pieces, then you, in some cases, you also need the transacting CRISPR RNA so that it forms an active form of this CRISPR RNA. So this loop is required for the interaction with the Cas9 protein. And well, when the complex is assembled, it targets to the foreign DNA and cleaves it, producing a double strand break. So, and of course, because the parts of the CRISPR array were obtained from the interaction with the virus, the small parts between repeats are specific to some viruses or some other foreign DNA that bacteria interacted with in the past. So these parts of the CRISPR array are specific to this particular foreign DNA that is familiar to the cell. So now let's review features of this system. So one of the features is that unlike the previous two genome editing systems, this one uses the guide RNA. And so it doesn't use the protein sequences to recognize DNA. Instead, it just utilizes the normal complementary interaction between RNA and DNA. So it is even more understandable to us. And so you do not require any specific code to understand how guide RNA targets a specific sequence. So in practical applications nowadays in genome editing, it might be cumbersome to use the two RNAs at the same time. So you need a mature CRISPR RNA and the transacting CRISPR RNA. So people came up with a solution for this by creating an artificial single guide RNA. So it combines the feature of the CRISPR RNA so that it has a recognition part, and it also has all of the hairpins and loops so that it is recognized by the Cas nuclease that produces the cuts themselves. So a small question for the audience is, why don't Cas proteins cut CRISPR arrays? Because, well, they should be specific for them as well.
Because of hairpin, they have hairpin, and its hairpin recognize palindrome and change conformation the following way, where guide RNA cannot sit by complementary to target, and Cas9 cannot cut CRISPR itself.
Yeah, all right. So yeah, and also the targets of the CRISPR-Cas systems, they have to comply to a very specific set of rules, and so not everything can be a target. And valid targets, they have to have a very specific motive beside them, and this is called the protospacer adjacent motive. And well, targets. So protospacer, here's the target sequence that need to be cut, and this motive, it is required for the target to be a valid target. And this protospacer adjacent motive, it depends on the Cas system at hand. So there are quite a lot of them, and well, maybe it is somewhat of a nuisance for the practical use, because it narrows down the available spectrum of target sequences. Now let's talk about the protein part of this system, and the most famous one. It utilizes a system that is used in genetic engineering, is the one that utilizes a single protein called Cas9, and it is one of the CRISPR-associated proteins, and it is able to cut both DNA strands at the same time. So it has a very specific domain structure, and two of these domains within this protein, they are required for cutting DNA strands. So one each. So one of these domains, they cut one DNA strand each, and one of the domains is required for RNA interaction. One of them is required for the protospacer adjacent motif interaction, and well, so later we'll see how you can manipulate some of this structure of the CRISPR-Cas9 protein to achieve different goals. And yeah, as I mentioned, basically, well, this type of defenses, they were evolving separately in different organisms, and that means that there are a lot of CRISPR systems out there. And it is also true for restriction enzyme systems, because, well, we know quite a lot of restriction enzymes that are functioning in different bacteria. So that means that there are a lot of different variations of arrangement of, well, what proteins do what, and what kind of protospacer adjacent motifs they are targeting, and what kind of proteins are required to produce the actual cut, and what is cut even. And so yeah, the basic system comes from Streptococcus pyogenes, and it requires, in natural circumstances, it requires the CRISPR RNA and the trans-CRISPR RNA. And the cool thing about it is it only requires one nuclease protein, and the protospacer adjacent domain is the well, two nucleotide sequence GG, and that is separated from the protospacer by one nucleotide that can be just about anything. And it produces blunt ends, so no overhangs. That may be a bad thing, by the way. So you cannot readily introduce something if you want to. So of course, there are some other widespread genome editing CRISPR-Cas systems right now. Well, at least one of them is called the Cas12a and CPF1. And the thing is, it's, as it is in the natural circumstances, it requires only one RNA, which is really cool. The protospacer adjacent motif is different, and it produces sticky ends, which is a great feature because you can use, well, sticky ends to introduce some kind of a insertion with the compatible sticky ends. So in this case, you may not only produce some mutations by the non-homologous end joining, but maybe introduce a very specific insert into the cell. And of course, if there are a lot of bacteria that possess the CRISPR-Cas systems, and they, well, vary wildly in their features, it is a very cool idea to mine the metagenomic data sets and genomic data sets for CRISPR systems. Because if you find one really cool CRISPR-Cas system, it may be a source of a great benefit for the field itself. So you can maybe use it in a specific subset of experiments of genome editing and so on and so forth. So it's probably a great idea to go about mining for new CRISPR systems. And yeah, so that is one of the applications of bioinformatics here. And it turns out that, well, people quite often find some new CRISPR-Cas systems, of course, and some of them are really strange when compared to the most basic ones. For instance, this one that uses the protein Cas13a is used, well, people are suggesting to use it for diagnostics because its behavior is very strange compared to the Cas9 from Streptococcus. And the idea here is that this protein, it cuts RNA, but it also causes a collateral damage, meaning that if it recognizes something in the solution, if it recognizes some kind of RNA in solution, it will start cutting, well, some other RNA species in the same solution, so without any differentiation. And it may be a great feature because if you put in the same solution some kind of fluorescent RNAs, And if this Cas13 protein goes rampant after recognizing the target, this special fluorescent RNase will also be destroyed. And it means that it can be used as a diagnostic tool. So, for instance, if you have some kind of a viral RNA in the solution and it is recognized by this system, you can easily detect it by the absence of the fluorescence after the recognition event. So yeah, and in the same paper, people described some cool features about it is that it is highly specific and quite robust. That it can differentiate even between very closely related strains, and also it is quite field conditions friendly, because in some countries that in the poorer countries, when you cannot afford to have a laboratory in every village, you may want to use this type of diagnostic system to find it in the field, to find out whether or not they have people have this virus, or maybe, I don't know, they contain this virus in some natural reservoirs. All right, so to compare CRISPR-Cas with the other genome editing techniques, it is quite easy to engineer and use, because in this case, you do not need to create a new protein every time you want to edit a new target. And it is a very cool feature, because at the same time, if you want to target many things simultaneously, you do not need to put several proteins with different recognition domains. You can just utilize multiple single guide RNAs and you're good to go. But yeah, maybe it's not such a great idea to use the multiple guide RNAs at the same time, because the off-target effects will also stack at the same time. So you will have more off-targets and possible cytotoxicity because of this. But yeah, the possibility is there at least. So if you want to switch target, it is quite easy. And single guide RNAs are also very, very easy to synthesize. So you only need to engineer the 20 nucleotide recognition sequence, and that's it. So you do not need to learn about some kind of code for tail nucleases or zinc finger nucleases. So yeah, it is quite user friendly compared to other genome editing. So, and now for some examples and practical uses of genome editing from recent news also. So one of the examples is it relies on your knowledge of the Mendelian genetics. Hopefully you all know that, well, diploid organisms, they contain two alleles. Alleles are independently split between gametes, so that during fertilization, alleles are randomly coupled. So you get quite robust proportions of progeny during your crossing experiments. And well, the underpinning of this process, of course, is the meiosis, that during which the separation of chromosomes happens. So diploid couples are split between the two daughter cells, and it turns out that this separation process is not always fair for both of the daughter cells, and there exists some process, some phenomenon that is called the meiotic drive, and this phenomenon is it's a phenomenon when some of the progeny during the cell divisions, they get one of the chromosomes preferentially. So it means that the separation is not exactly equal, and in some cases it may turn into a very skewed proportion of phenotypes in the progeny when you do the crossing experiments. And the mechanism of this maybe is that well, it is in plants. It may work as that they have some chromosomes have some kind of heterochromatic regions, and some of the forms of these organisms, they have a specific kinesins that bind to these heterochromatic regions, and so that they, well, these chromosomes with this knob of heterochromatin, they are transmitted preferentially. And in some other cases, meiotic drive can lead even to more drastic changes. So in this case, it's I don't really remember the phenotype of this, but in case of this example in flies, the driver alleles of some specific gene can lead to a non-segregation of Y chromosome, and it can lead to a shift in sex ratio between in the progeny. So, in the long run, it may have quite a drastic implications for the population. So if you change the ratio of gametes, so it may lead to the shift in the, well, some phenotype proportion in the population. And it turns out that the similar effect can be achieved with the genome editing. So we talked about those. So in case of the Cas9 system, you only need Cas9 protein and the guide RNA to achieve the editing event, right? So if you insert this type of cassette into the host genome, this guide RNA that is inserted, it's going to be targeting the same locus that the cassette was inserted. So in this case, you will get a very strange situation, because, for instance, you can see that in the genome, there is one allele that is quite normal. Better here. So there is one allele that is quite normal, and another one, it contains this cassette of the Cas9 and the guide RNA. So these two genes are quite able on themselves within the living organisms. They are able to modify the second allele. So you can see that it can result in a very extreme deviation from the normal proportion of 1:1 between alleles. So in the most dramatic circumstances, that can lead to the disappearance of the second allele at all. So yeah, and this type of phenomenon, this type of experiment, I don't really know. It is called the mutagenic chain reaction. So this Cas9 cassette is inserted at a certain locus that targets the same locus. Yeah, and it can lead to, well, to the fact that all of the progeny of some cross that contains this gene drive mutagenic chain reaction cassette, all of the progeny will carry the same allele. So in the normal Mendelian inheritance, only half of the chromosomes will have this cassette, right? So one of the normal chromosomes comes from here, modified, or maybe, I don't know, mutated chromosome comes from here with the mutation. So you can see that individuals are heterozygous. But in case of the mutagenic chain reaction, the normal chromosome will be modified. And it turns out that, yeah, in this case, the recessive trait will be apparent in the progeny in the first generation. So in the original article on the mutagenic chain reaction, it was actually done, and yeah, people see that in the first generation, people achieved first of all, it shouldn't be possible to even have the yellow individuals, the yellow female individuals in the progeny, but it turns out that there are quite a lot of them in the progeny. So it was quite strange to even see them in the result of the cross. So yeah, so it turned out that the thing actually works. The system of mutagenic chain reaction actually works, and in population it can lead to a very dramatic effect that altered gene is always inherited, and the allele will spread like a wildfire in the population, because yeah, it is certainly not a normal inheritance. In normal inheritance, it will disappear at some point, maybe, but yeah, if you use the gene drive allele that can propagate itself with the genome editing, it will always stay in the population. So it turns out that it's not always the ideal case. So this new allele does not always modify the second one. So there are some other implications here, and at least in some cases, the new allele that provides the gene drive, it does not retain. It is not retained in the population. So it depends on the fitness cost. So if it is too expensive to maintain this allele, maybe it will disappear at some point. And also, if the conversion rate is not so high, so that the second allele is not modified enough to be for the gene drive cassette to be retained in the population. So people actually try to utilize this type of the technology. So yeah, it is quite dangerous, as you can imagine. So people in the original article, they even warn about so it can be dangerous if you use it in the natural population. But people are trying to find the application for this, and there are some actually. So at least one of them is reliant on the sex-specific splicing events in the Diptera, so in flies and mosquitoes. So depending on the sex of the individual, one of the transcripts is spliced differently. So, and there is some specific intron that is required for this. So you can introduce some kind of an intron that is spliced differently in females and males, and it will result into different transcripts in the different individuals. So it turns out that you can use this strategy to fight against mosquitoes. So you can modify the female version of double sex transcript to destroy the malaria transmitting transcripts. So you can introduce the same gene drive cassette. So in this case, it turns out that mutants that are produced by introduction of this gene drive cassette are phenotypically aberrant males. So they should be females, but they are not normal females, so they cannot reproduce at all. And well, homozygous mutants are sterile, means that well, no progeny for them. And heritability, oh yeah, see it here. So the heritability of gene drive cassette is actually quite high in this case. Transmission was almost 100%. So, and even heterozygous organisms are quite a bit less fertile. So you can use it to counteract the reproduction of mosquitoes that carry some diseases. And yeah, as you may expect, if it is transmission is quite high and the fitness cost is, well, actually just about everything, the population dies in a very small number of generations. So isolated population at least. And yeah, it is actually quite a hot topic because well, malaria turns out to be a huge deal in at least some countries, and there are quite a lot of cases worldwide. So, and the progress in the fighting against malaria is not so hot right now, because it was really great at some point, but recently it nearly stopped because, well, there is only so much you can achieve with maybe the infrastructure measures and something like this. So there are some projects that are fighting against the malaria, and at least one of them, in one part of this project, gene drive is planned to be used in Hawaii to save the endemic bird species, because they are quite susceptible to these mosquitoes. And in order to save this disappearing species, endangered species, people are actually planning to use this kind of a mutagenic chain reaction. And it turns out that similar technology that produce sterile insects are already approved. So people are already using them, and despite how wild it sounds to us, maybe, people are using this, well, similar strategies to produce the sterile females, so that they stop the population growth of these mosquitoes. And one of these technologies created by Oxitec, and here the idea is actually quite similar in the sense that there is a also sex-specific intron that is spliced normally in males, but is spliced aberrantly. No, it is spliced aberrantly in males and normally in females, so that well, in female individuals, some malicious, not malicious, but some bad protein accumulates in the organism, and well, they are unable to reproduce normally. So yeah, and this thing is actually approved right now. So in this August, the pilot study was approved in Florida Keys, and well, the day before yesterday, another pilot study was approved in Brazil. So so there was another pilot study in Brazil earlier. It was successful, so they widen it right now in scope. And it looks like basically a pill. So they even called it just add water, something, I don't know, just add water to this pill, and basically it will reduce the spread of these mosquitoes that carry diseases. Also, the same idea can be applied to, well, mammals, of course, and there are quite a lot of bad mammals that maybe need to be population controlled. But the bad thing is that gene drive requires homologous recombination, and mammals mostly use the other type of the DNA repair. So there are ways to counteract it, and one very curious way to do this is to exploit the fact that different DNA repair mechanisms are utilized in different stages of cell cycle, and in particular in the different stages of the gametogenesis. It turns out that in female gametogenesis, there are some stages where homologous recombination is quite ubiquitous, so it occurs a lot, and you can target your genome editing events to these particular stages so that you get the gene drive instead of just a double-strand mutation, which leads to nothing basically. So, and it turns out that if the gene conversion happens during this specific part of the oogenesis, the conversion effects are happen quite often. So yeah, and it is also planned to be used in the field, and here it's used in another part of the world, in particular in New Zealand. And maybe know that quite a lot of local species are endangered because people introduce unnatural species there, like cats that like to hunt small and vulnerable local species, and people try to plan to use the gene drives to kill non-native predators. So they also use very physical ways to dispose of them, but gene drive is one of the possible application of gene editing there. So, and they even plan to use a more advanced version of gene drive, which is called the DAISY drive. And the idea here is that instead of modifying only one locus, they use several elements, and one of these locus, it drives another one, and this one in turn drives another locus, so that this type of genome modification results in a very explosive effect, so that it happens very, very fast and possibly eliminates the population, and then it disappears also quite rapidly, because once it spreads quite a lot, genes can no longer drive each other. So, and in this case, it's not indefinite and not quite as dangerous as the vanilla gene drive. So yeah, and people also try to, of course, because of this implication, gene drive seems quite dangerous, because it can also, in the simplest form, it can spread indefinitely and globally. So people, of course, try to somehow measure its effect on the population, but it turns out that the findings are quite unnerving, because gene drive cassettes can easily escape control even if the conversion frequencies are very low. So if you are not very cautious about using them, you may end up in a lot of problems because maybe it will spread to a related species and cause a lot of environmental damage. So yeah, and we still have a couple of minutes for the last part of the genome editing application. So, and here we'll talk about the first human embryo genome editing. So the first one published was performed on the triploid embryos. So there is no way they are they can be transformed into normal individual later. So they just die, and they cannot give rise to a normal person. And they tried to introduce mutation in a familiar to us gene of the hemoglobin beta. So yeah, they used guide RNAs targeting this gene, and they also used some donor oligonucleotides to introduce mutations. So here it is edited with a single-strand RNA, and the effectiveness of editing was not, well, quite high. So you can see about 50%, and recombination events with the off-target gene were also noticeable, and editing percentage was very low. So as it was, I think that in this particular paper they concluded that it's not applicable as it is to human genome editing. But yeah, so there are some modifications should be in place for it to be used clinically. So here are some off-target activity in this particular study. So they found that there are some other sites that are modified by the same single guide RNA, and some of them are, well, quite different from the intended target. So of course, the homologous gene is also affected by the same modification, despite the fact that it has, well, quite a lot of substitutions compared to the intended targets. So, but of course, it was quite a while ago, and people still try to do some experiments on the human embryos, and they are testing different strategies once again for genome editing, and of course different stages for injection. So what it's like in the example with mice and the different studies of the oogenesis. So people try to inject the Cas9 protein and guide RNA in different studies to maybe achieve different effects and in editing. Now, in this particular study, people targeted the gene that causes cardiomyopathy. It's a very interesting disease in the sense that it causes sudden death in very young and well, otherwise healthy individuals, and you do not really know until it strikes. So quite a bad disease. So you probably want to somehow eliminate it, especially considering the fact that it is autosomal dominant. And so genome editing would be able to rectify this errors in the gene. So yeah, they targeted a very specific indel, so to well, get it normal again, to get the gene normal again. And so once again, they used they utilized our knowledge about the cell cycle in gametogenesis to introduce guide RNA in the very specific moment where editing may produce better results. So when they introduced the editing proteins and RNAs during the S phase infection, during the S phase of division, they had very many mosaic embryos, which is not very cool, and had a very low effectiveness at all. So a lot of the resultant embryos were not even well, modified at all. So but next they tried to introduce the editing event in another phase, namely the meiosis II, and they got sufficiently better results in the sense that they no longer had any mutated individuals. So in the previous case, they had some unedited embryos, and in this case, they only had, most of them were edited, and some of them were mosaic, but the percentage is, percentage is, wait, something's not wrong here. That's right. So yeah, it says that it should be uniform, but I don't really understand. Oh yeah, it says the different types of modification, not mosaics. So yeah, they did not really have any mosaics here, which is really cool. So yeah, and embryo morphology after this injection was not in any type, was not at all aberrant. So probably embryos would be normal after this injection. So it does not disrupt any development. So, and of course, you probably have heard about the scandal about the modified children that were modified in a familiar to you gene CCR5. So this mutation was introduced into them, and that the names of these children are Lulu and Nana. So the experiment was conducted in China, and the people claim that they contain no off target, but we don't really know. And I don't really remember the exact punishment that was done to this person who conducted the editing. But so now we have a precedent, at least, when modification was applied to some people that actually live and that are not embryos somehow. So, and of course, in a smaller scale, CRISPR can also be used for modifying a small isolated population of cells within the organism, and there are some clinical trials about this. So, so there are examples of using CRISPR against melanoma. There are examples of usage of CRISPR for CCR5 disruption once again. So, so in this case, you do not need to modify the entire organism; you just modify the stem cells. So, and in this particular case, it's not a huge success because the efficiency of targeting was slightly low and no remission was achieved. So, but at least they had no adverse effects. And as for the legislation, most of the time, in most of the countries, the genome editing for therapeutical purposes is not exactly permitted right now. So you can see that it is only permissive. The legislation is only permissive in Mexico and China, as it is. Maybe in other countries too, because they are not listed in this case. Yeah. So, and of course, as I mentioned, so we know the domain structure of Cas9 proteins, so you can modify it, and you can modify the guide RNA to achieve some other types of targeting aside from double strand cleavage of DNA. And you can use it for transcriptional regulation, like for transcriptional repressors or activators. You can splice a fluorescent protein to the Cas9 for some analog of fish. You can somehow use the somehow use the Cas9 protein to introduce some epigenetic markers and so on and so forth. So it's also applicable to guide RNA. So you can use some fluorescent RNAs. You can use some other protein binding motifs to ensure that other proteins interact with the guide RNA. And yeah, so there are also examples of this kind of applications for CRISPR. And in some cases, CRISPR can also substitute a very widespread and traditional applications of bioinformatics and gene expression studies, because while high throughput RNA sequencing or microarray can achieve a very systematic analysis, but they do not know what actually caused the difference between samples. And CRISPR-Cas libraries, they can give you this. So in some cases, CRISPR can achieve studying the causality in experiments of high throughput profiling of genes. So in this case, you see that if you disrupt a lot of genes and you systematically analyze the aftermath of this disruption, you can see that for a lot of cells, for a lot of cells that undergone some particular genome editing event, so you can actually tell what caused it. So in this case, you do not just observe the expression differences, you can also say what exactly caused them in a controlled environment. So yeah, that's about it for genome editing lectures. So if you have any questions, now is the time. All right, thank you all for your attention then. I'll see you in a couple of weeks, I think.
Thank you.